the plants and that you want to do plants, I think we can actually see now a little bit a poster of Petya Stoykova. Petya Stoykova, are you there?
Hello, I'm here.
Hi.
I was just wanted, yeah.
Don't be shy.
wanted to take the word first to say hi. I'm really impressed and thank you so much for organizing this conference. This is my first aquaphotomics conference. A lot of information, quite new for me because I'm actually a molecular biologist, but I'm really impressed for the applicability of this method of aquaphotomics that can be used in so many fields of the science. And also I want to thank to make this poster reality, and if possible, I could try to share my screen, or you could share it. I will be really thankful to people that presented their lectures on materials and detection of water interactions with different materials, because our poster worked in collaboration with Professor Stöykova's group is related to detection of water structures when hydrophobic molecules are dissolved. So I will be really thankful to any comments of people that deal with hydrophobic molecules and interaction with water.
Hydrophobic. Well, hydrophobic, yes. I'm interested also in hydrophobic components. I want to just accentuate something that you said, because this is something that often that I was really shocked when I met you first. And so you were asking me, and that is something I see with not only molecular biologists. Say, you use only one method. I have to learn so many. I have so many folders and I was like, oh God, that's so much work. So thank you for that. That was really interesting to know. So anyone has experience with hydrophobic pollutants here?
Just to mention something else. So this is a part of our huge experiment. We, with Professor Stöykova's lab, we have a lot of data, a lot of spectra, and on plants that are contaminated or stressed with these contaminants. And this is this poster includes only data on these two hydrophobic molecules dissolved in water, but with added organic solvent DMSO. So I will be really thankful if any comments on what really is happening in this environment, water plus DMSO. This is my first question from chemists or some other people involved in that. And second, if like Vlada said, if we want, for example, want to obtain models for detection of these components in the environment samples, not mainly in water, but for example, their pollutants, soil pollutants. So is there a way by adding some adjuvant molecules, like for example, for me, like as a molecular biologist, I perceive DMSO or ethanol like adjuvants that influence additionally the water structure and help to decrease the limit of detection of these molecules.
Yes, that's a very good question.
Is it correct to that we added this DMSO? Is it okay, this percentage? And also, is there a room to proceed with these investigations by varying the percentage of organic solvents in order to search a way to decrease the limit of detection of such molecules in environmental samples.
That's really, yes, I'm listening.
Petya, I'm happy to see you, and I'm glad that you like it. Could you please introduce your poster? Just a very short, what is the work? And I found, I didn't know about your work and Professor Inui's work until we met. And I think we were shocked both sides because it was so interdisciplinary and out of discipline. But at the end, I think your work is remarkable. Exactly, if you tell the road that you walked, so it will be a very good example for everybody. As we are people from so many different disciplines, and it will be a very good example. Yeah.
Yeah, that's true. Thank you so much for the questions. And in the recent years, it's very common for me to use the sentence for the first time. For the first time, it happens. Always something happens for the first time for me. So in 2017, I went to Professor Inui's lab to work on environmental pollution. So we used genetically modified plants as an alternative to detect organic pollutants by these genetically modified plants as an alternative method to instrumental methods. And then during my work there, we just imagine that it will be interesting to start to observe what is going in plants when a pollutant is introduced. Can we use aquaphotomics to detect this pollutant in vivo in plants? So we started from a very simple experiment and then repeated several times by varying different treatments. So I might say that based on preliminary reports received from Jelena, we have a very good discrimination between polluted and not polluted plants. Until now, we tried this experiment with only one plant species, with zucchini. As I might say, model plant that is able to accumulate these organic pollutants to upper parts into leaves. So that's why we our idea was not only in vivo, but we wanted to also try to detect these pollutants and to see is there a difference in the limit of detection when we try this method in vivo, in vitro, and just in water environment. And what was striking and great for us, for example, if you see in the poster, limit of detection of pyrene, this hydrophobic compound, in water environment is in the range of ppm. And what I remember from our preliminary results and from Jelena reports was that somehow in vivo environment, the plant cell, because we measured the leaves of the plants while it was growing on soil. So in this environment, we have a lot of molecules, active enzymes and DNA, all these apparatus is actively working. So the limit of detection of these compounds was much lower, which means that somehow this living environment and stress response of plants somehow zooms boost this effect of this hydrophobic xenobiotic molecule to the living organism.
Yeah, that's beautifully said. Accentuate. Because water, I mean, we usually say sensor and amplifier. Amplifier signal. Thank you, Petya.
Can I will just, I think this is a very good place. To me, your problem and your scientific problem looks differently. It is not quantity. It is not limit of detection. Of course, it is. So we measure something that is not measurable, cannot be measured, etc. But I think the problem is that this has been solved, and it shows that it could be solved by the water. Yes, thank you. Is something that was undoable until now. So we want to know the how do we have heavy metals in the soil, or we don't. That's the problem, right? And you want to solve this problem by growing plants in the soil for 1 week, and you monitor these plants, and you see how the water spectrum changes for a week, and then you can measure if there is a heavy metal in the soil. This is how I see the problem. Am I right?
Yeah, that's true. And in this particular case, in the case of hydrophobic molecules, I think we are still not quite familiar what's really going on with these water shales. For example, for me, it was very interesting why we have this good linear correlation for the concentration of pyrene, where with increasing the concentration, we have this linearity. Pyrene is a hydrophobic compound, and it is not dissolved. The percentage that is dissolved in water is very, very low. So by increasing the concentration, we have this dispersion. We have these particles consisting of pyrene molecules. So since it's not completely dissolved in water, we have these particles. So the water molecules that are involved and affected by pyrene concentration should not be in linear concentration depending on the pyrene concentration, but still we have this this influence, linear influence of pyrene concentration.
I think that was puzzling.
When I look at our programs, so we are not. It is a linear combination, but you have a spectral pattern that is changing in a linear way, right? It is there are different water species that are changing according to the concentration of pyrene, and I don't think it is only the pyrene that is changing. But there are so many other things in the plants that are changing accordingly. So that's why you can have a very beautiful correlation between many other things influencing the water. So you're looking at the whole system of the plant. And this is, I think this is the beauty of the approach. So it's absolutely unconventional way of solving the problem. Instead of going and measuring heavy metals in the soil, you just, you grow a plant for a few days, you measure how the water is changing in the plant, and then you have the model for the pollutant. So it's, this is the beauty, yeah, I think.
And that is the answer to what Vladimir commented just on the chat. Can I hear the question from Zoltan Kovac?
Yes, thank you. Actually, you already partly answered while I was looking at your spectra, and I'm not familiar with this kind of materials, with hydrophobics, but you mentioned that they are not very well dissolved. The solubility is not that good, right? So it might influence the spectra, and I think this is what you are showing in the different ranges, that the there is an offset and slope difference. So I would be very interested to see your loading vectors and regression vectors. But why I'm saying this is because I think if this is the first measure, then you might be able to choose only few wavelengths to really predict the presence or concentration of these materials. And with that, you could suggest a very, very cheap instrument. So we don't need probably very wide range of spectrum, but one or few selected wavelengths probably can do the job. And I don't know what kind of pretreatments you have tried so far. Have you tried, for example, detrending?
Yes. So, Zoltan, the results that are presented here are I did a combination of different treatments. We have like a table of these combinations that we tried, and these are the best of the pre-treatment that work on these particular regions. We also varied the different regions. So what so you're absolutely right that what we calculated here is based on the slope and on for the case of pyrene. But because the point is that there is a difference in the structure of these compounds, and this structure, I think, in the water gives rise to self-organization. So this self-organization, basically, owing to that, pyrene makes structures which scatter light, and that is the basis for the measurements of pyrene, I think. And pyrene shows beautiful spectra. You know, you have a baseline changes from visible. It's basically used in based on fluorescence also, right? Petya? It's fluorescence compound. So you have a beautiful, you know, baseline to play with. Dieldrin is not like that. So in the case of dieldrin, we rely definitely on chemical changes. And you can see, it's not so beautiful. I mean, it's not, okay, it's not perfect, but I also want to make a pyrene could be also predicted based on the totally raw spectra with the basically same, and only, I think there was like 3 wavelengths. So you would need maybe just, you know, baseline, nothing else.
Perfect. Thank you very much.
Thank you, Zoltan. Oh, Felix.
I have a question. Yeah. Hello.
Yeah.
Hello, Milano. Hello, Jelena.
Hi, Felix.
Greetings from Switzerland.
Welcome.
So this was a nice presentation about the use of aquaphotomics with regard to environmental toxicology. And I think it's an important application because really we have a problem with pollutants in water. And one thing I was not clear about the approach was, did you try to mix these 2 components and create then the PLSR models, or did you just create a model for each component separately?
No, so in this case, this was this is the first work that we are presenting. So in this case, we did not mix components. So they are in totally separated solution. One is solution of dieldrin, one is solution of pyrene with DMSO. So we use DMSO to dissolve the compounds, but the components are not mixed.
That would be interesting to see what is happening when we mix different components.
Yes, absolutely, absolutely. So in regard to that, this is very similar. We had recently a study on which utilized DMSO to dissolve some other compounds like NAD, NADH, and the NAD+. We were interested in those compounds because of the ratio is indication of oxidative stress. So the point is that one of the compounds were not, I could not develop a model. For the other compound in the same solution, I was able to, let's say, predict rationally, relatively good model developed. But when I modeled ratio of NADH, pardon me now if I'm wrong about which comes, which is which. The point is that when we tried with the ratio, which it basically influences that is what we are going to see in the water, the synergetic influence of all compounds. So when I modeled the ratio, I had a good model. So it is in the what you get in the end. This is what you want to model based on that.
Yeah, but I would also expect that there are synergistic effects happening, especially since here in Switzerland we have dozens of chemicals in the water, including glyphosate. So maybe it would be very interesting to create also the analysis with glyphosate.
Yes, Felix.
Would love to
Just
see this Aquaphotomics glyphosate toxicology study being done.
Yes, just do it. I mean, glyphosate is a big problem. Absolutely.
I